Class 11. In today's class, we shall discuss proteins. When we look at a cell through a microscope or analyze its electrical or biochemical activities, we are actually observing proteins. Because proteins constitute most of a cell's dry mass. They are not only the building blocks from which cells are built, they also execute nearly all cell functions. Thus, enzymes provide the intricate molecular surfaces in a cell that promote its many chemical reactions. You know of some proteins that remain embedded in the plasma membrane to form channels and pumps that control the passage of small molecules into and out of the cell. Some other proteins are also there which carry messages from one cell to another. Some act as signal integrators that relay sets of signals inward from the plasma membrane to the cell nucleus. Some others are tiny molecular machines with moving parts. For example, kinesin. It propels organelles through the cytoplasm. Topoisomerase, you have heard the name. It untangles knotted DNA molecules. Other specialized proteins act as antibodies, toxins, hormones, antifreeze molecules, elastic fibers, ropes, or sources of luminescence. However, from a chemical point of view, proteins are by far the most structurally complex and functionally sophisticated molecules known. The shape of a protein is specified by its amino acid sequence. A protein molecule is made from a long chain of amino acids, each linked to its neighboring amino acid through a covalent peptide bond. Many amino acids form a chain of amino acids. Therefore, proteins are also known as polypeptides. Each type of protein has a unique sequence of amino acids. Many thousands of different proteins are known, each with its own particular amino acid sequence. However, before entering into the details of protein, their structures, their functions, etc., I think it would be convenient for you to, if I discuss the structures of a few amino acids and the way they form peptide bonds. However, you know that amino acids form polypeptide chains, and those chains are further constrained by many different set of weak non-covalent bonds that form between one part of the chain and another. Non-covalent bonds, uh, mm, covalent bonds, ionic bonds, etc. Non-covalent bonds means hydrogen bonds, ionic bonds, van der Waals attractions, etc. These are weak bonds, but many weak bonds together can act as a strong bond which can hold two regions of a polypeptide chain tightly together. The stability of each folded shape is therefore determined by the combined strength of large numbers of such non-covalent bonds. You have seen in the case of DNA and RNA that huge number of hydrogen bonds hold the building blocks of these nucleic acids, that is the nucleotides, together, and the two strands are also held together by the hydrogen bonds. And some you have heard the hydrogen bond is also formed for asymmetric distribution of electrons between oxygen and two hydrogen atoms, and for this reason the oxygen part remains slightly negative, and the hydrogen parts whose electrons are drawn into the oxygen part, the hydrogen parts become slightly positive, delta positive, and these are called polar molecules because of this. In spite of being neutral as a whole, they have electropositive or electronegative sites in different regions. So these form the hydrogen bonds. As a result of all these interactions, each type of protein has a particular three-dimensional structure, which is determined by the order of the amino acids in its chain. The final folded structure is called conformation, adopted by any polypeptide chain, is generally the one in which the free energy is minimized. Protein folding has been studied in a test tube by using highly purified proteins. A protein can be unfolded or denatured. That means its conformation is changed, its folded form is changed. It is now denatured by treatment with certain solvents like detergents, etc., which disrupt the non-covalent interactions holding the folded chain together. This treatment converts the protein into a flexible polypeptide chain that has lost its natural shape. But when the denaturing solvent, that is the agent used for denaturation, is removed, the protein often refolds spontaneously. That process is called renaturation. We have seen it in the case of DNA. Protein renatures into its original conformation. Although a protein chain can fold into its correct conformation without outside help, protein folding in a living cell is often assisted by special proteins which are called molecular chaperones. Mind it, molecular chaperones are the special proteins which assist in folding up different proteins in a cell. Chaperones are vital in the crowded conditions of the cytoplasm. ভাবো তো একবার, সাইটোপ্লাজমের মধ্যে বেশিটাই তো প্রোটিন, কত রকমের অঙ্গাণু আছে, কত প্রোটিন আছে, সাইটোপ্লাজমিক যে নেটওয়ার্কটা আছে সেগুলোও প্রোটিন দিয়ে তৈরি। আমি একে একে আলোচনা করব, হয়তো তোমার বইয়ের সঙ্গে পরপর হবে না, কিন্তু সবগুলোই আলোচিত হবে। যাই হোক, chaperones simply make the folding process more reliable, and proteins have a wide variety of shapes, and they are generally between 50 to 2000 amino acids long. Large proteins generally consist of several distinct protein domains. Domain means area, which are domain means structural units that fold more or less independently of each other. এবং সেই জায়গাটা একটা ফাংশন আছে আলাদা। When these three-dimensional structures of many different protein molecules are compared, it becomes clear that although the overall conformation of each protein is unique, two regular folding patterns are often found in parts of them. The first folding pattern to be discovered is called the alpha helix. It is found in the protein alpha keratin, which is abundant in skin and its derivatives, such as hair, nails, horns, and one very peculiar thing I can mention for you, that the protein keratin is formed by all vertebrates. It is the chief structural component of hair, scales, horn, wool, nails, and feathers. The black rhinoceros is nearing extinction in the wild because of the myths prevalent in some parts of the world, especially in China, that a powder derived from the horn of a black rhinoceros has aphrodisiac properties. মানে এটা যে সেক্সুয়াল ড্রাইভ অফ হিউম্যান বিইংস সেটাকে ইনক্রিজ করে, এনহ্যান্সড করে। বাট ইন রিয়েলিটি the chemical properties of the horns of these innocent animals are no different from those of powdered bovine hooves or human fingernails. তোমার নখ কেটে যে কেরাটিন পাওয়া যায়, গরুর মরে গেলে তার খুর থেকে যে কেরাটিন পাওয়া যায়, আর ওই গন্ডারের খড়গতে যে কেরাটিন পাওয়া যায়, সেম। কিন্তু এই ভুল ভাবনা থেকে এই গন্ডারগুলো শেষ হতে বসেছে। Widely diverse products such as enzymes, hormones, antibodies, the lens protein of the eye, transparent lens protein of the eye, feathers of birds, spider webs, যে মাকড়সা যে জাল বনে, spider webs, milk proteins, antibiotics, mushroom poisons, and a myriad of other substances having distinct biological activities are found in proteins, different kinds of proteins.